Hi, this is Ayan and welcome to another new session on my channel. This is the last part or the last class of photosynthesis. Alright, so let's begin the class. This is going to be a very short class. If you have any doubts, if you have any questions, you are supposed to ask that. I will be answering that because there's nothing much to teach. It will be a class of 40 minutes approx. Right. So please just let me know in the live chat whether are you online or not. Are you with me or not? Just show or just tell me in the live chat whether you're with me or not. Right. Are you with me on the live stream? Are you with me on the live stream? Okay, nice. So let's begin the live stream. So today we are going to talk about, today we are going to learn about the concept of dark reaction. So the concept of dark reaction. So what's the better term that we should use for dark reaction that I told you? What's the better term that we should use for dark reaction? I have told you in the previous class. Please do let me know in the comment section. What is the other name for dark reaction, or what is the more appropriate name for dark reaction? Let me know. Then we will talk about the site of dark reaction, and then we will get a brief overview of dark reaction. Light independent reaction, very good. It's known as light independent reaction. So why it is known as light independent reaction? Because the steps of this reaction does not require radiant energy for its functioning, right? Light independent reaction, very good. Now can you tell me the site of dark reaction? The site of dark reaction. Can you tell me the site of dark reaction? The site of dark reaction is the stroma of chloroplast, right? The stroma of chloroplast. Right. And now basically what happens in the dark reaction, if we try to see, in dark reaction we take in the high energy intermediates that are produced in the light reaction, that is ATP and NADPH, and the carbon dioxide that is taken in from the atmosphere, very good stroma, and the carbon dioxide that is taken in from the atmosphere is reduced into what? C6H12O6. Now C6H12O6 can be so many other things. It can be galactose, it can be fructose, and so many things, but it is not. In case of photosynthesis, it is particularly what? It is particularly glucose, right? Okay, not a problem, not a problem. Even if there's a lag, not a problem, we can deal with it. Now what happens? Now this reaction, that is from CO2 to C6H12O6, this reaction is a reduction reaction. Right? So whatever I am saying, if you agree with that, please write a quick Y, and if you do not agree with that, write a quick N, And if you have any question that you are going to write and ask, please put a question mark beforehand and post it so that I can understand that okay, you are writing a question and you are going to ask a question, right? So this can help us in easy, fast and effective communication, right? Okay, now this is reduction. Now in this connection I said a few things in the previous class, that is reduction, whenever you have an anabolic reaction, that they will show anabolic reaction, anabolism or anabolic reaction, mostly reduction, and catabolism is mostly oxidation, right? Isn't it? So this idea is mostly, almost, these things happen, right? Now again, another thing that we discussed in the previous class that we should learn right now is that over here we have something, we have non-cyclic photophosphorylation and we have something known as cyclic photophosphorylation. These are the two types of light reaction or light dependent reaction, right? Now cyclic photophosphorylation produces what? It produces NADPH only, whereas non-cyclic photophosphorylation produces what? NADPH plus ATP. Yes or no? Right? Now, so over here from this we can infer what? There's an inference that we can draw from this place, that the number of NADPH produced, number of NADP, am I writing it right? Am I writing it right? Please follow the class. You never mentioned that I made a very big mistake. What mistake? Where's the mistake? Identify the mistake. That was a deliberate one. Identify. ATP is produced in both. Okay. Then what about NADPH? Not NADPH. So where NADPH is produced? In which reaction NADPH is produced? In which reaction NADPH is produced? Non-cyclic. Non-cyclic NADPH is produced and ATP is produced, and so in cyclic photophosphorylation we have only ATP produced, right? So basically over here I made a mistake. This is not the correct thing. Over here only we will have ATP produced, right? So over there we should have ATP only. Nice. Now from here we can draw a quick conclusion, that is both of the processes are producing ATP, but only one of the processes is producing NADPH. That means the number of ATP produced is obviously more than the number of NADPH. Yes or no? Right? Now with this basic idea we can dive into the the main idea of dark reaction. Now we discussed that dark reaction can occur in two ways. It can occur as a C3 cycle and then it can occur as a C4 cycle, right? Now first let's understand the C3 cycle and then we will go on with the further discussion. We will continue with the further discussion. So let's see the C3 cycle. So basically this is a C3 cycle. This is one of your C3 cycle, right? Now over here I would request you to ignore the balancings. They have tried, in this diagram it has been tried to balance the reaction by putting the numbers 3CO2, then 6 3PGA, everything, right? Just ignore those balancings. Okay, ignore this for now. So should I start with the C3 cycle? Should I start with the C3 cycle? All right. So now basically what's happening is that in the stroma of the chloroplast we have a molecule known as RUBP, right? RUBP. So let's zoom it in a bit more. Let's zoom in a bit more over here. So yeah, so we have a molecule known as RUBP. Now this RUBP, the full form of RUBP is ribulose bisphosphate. The full form of RUBP is your ribulose bisphosphate. Right. So ribulose is a molecule, and along with that bis means 2, and bisphosphate means there are 2 phosphates attached to the ribulose. So as you can see in this figure, this part is the ribulose part, and along with that there are 2 phosphates attached at the 2 ends, right? Are you following it? Have you understood whatever I told till now? Have you understood? Now, this ribulose is a kind of sugar. Now, in the previous classes, we learned that there's something known as ribose. So, ribose, which is a 5 carbon containing sugar. Now, this ribulose is a similar kind of a sugar which also contains 5 carbons in it, and it is similar to ribose. Okay, now there's a molecule known as RUBP which is present in the stroma of the chloroplast. Now, what is the full form of RUBP? RUBP is a molecule known as ribulose bisphosphate. Now, let's break down this name to understand what it is. So, there's something known as a ribulose part. Along with that, we have the term bisphosphate. That means bis means 2 and phosphate means the phosphate group or inorganic phosphate group, right? And that means there are 2 phosphate groups attached to something known as ribulose, right? So as you can see in the diagram over here, we have 2 phosphate groups attached at the 2 ends of something known as ribulose. Now, what is ribulose? So in previous classes we learned that there's a sugar, 5 carbon sugar known as ribose, which is present in the nucleotides, right? Now, ribulose is a similar kind of sugar. It is very much similar to ribose, and that means it is also a 5 carbon sugar, right? Now, to represent this, we have put 5 purple circles over there. See, over here we have 1, 2, 3, 4, 5. With 5 purple circles we have put in to represent ribulose. Now, ribulose is a 5 carbon containing sugar. And with this 5 carbon containing sugar of the ribulose, there are 2 phosphate groups attached at 2 ends of it, right? So this is the meaning of the name ribulose bisphosphate. Ribulose bisphosphate. Okay, nice. Now, the carbon dioxide which was absorbed from the atmosphere, now basically we are signifying the number of carbons over here with purple dots. Now, one molecule of carbon dioxide contains how many carbon atoms? How many carbon atoms? One molecule of carbon dioxide contains how many carbon atoms? 1, right? So hence over here in this chart we have signified CO2 with one purple circle, right? Now over here comes the RUBP taken in from the stroma, and along with that fuses or reacts the carbon dioxide taken in from the atmosphere, and they react to form something known as this structure. Okay. This is some 6 carbon containing intermediate. So are you with me? This is an intermediate. Now if you count over here how many, and again ignore this balancings, over here how many carbon atoms we have? 1, 2, 3, 4, 5, 6. So basically this intermediate is 6 carbon containing, right? So it's very much evident we had a 5 carbon containing compound, that was RUBP, and along with that we combined one molecule of carbon dioxide, and carbon dioxide is 1 carbon containing, one molecule contains 1 atom, and hence this 5 carbon plus 1 carbon makes up an intermediate known with 6 carbons, and the phosphates of RUBP are kept intact, right? And this reaction is catalyzed by one enzyme known as Rubisco. This is the important enzyme that catalyzes the reaction. The enzyme is known as Rubisco. Right. Now few words about the Rubisco. Rubisco simply means RUBP carboxylase, RUBP CO. Now this C stands for carboxylase and O stands for oxygenase. C stands for carboxylase and O stands for oxygenase. Now if we focus on that a bit more, now this C means what? Carboxylase, and O means what? Oxygenase. Right, carboxylase and oxygenase. Now carboxylase means what? Carboxylation is a reaction where carbon dioxide is incorporated or added to one molecule. Suppose we have some molecule, along with that carbon dioxide gets added, and then there's a product. This reaction is a carboxylase. And what is an oxygenase reaction? Oxygenation is molecule plus oxygen forming product, that is an oxygenation reaction. Now this enzyme Rubisco, which is essentially RUBP carboxylase oxygenase, can perform carboxylation as well as oxygenation of RUBP. Over here it causes carboxylation in presence of CO2, it causes oxygenation in presence of O2. Now since we have presence of CO2 over here, it will cause carboxylation or through its carboxylase activity, and it will cause incorporation of the CO2 molecule into the RUBP molecule. Over here the molecule is basically our RUBP. Are you clear till this? Are we clear? Have we understood till this part? What is RUBP? What is Rubisco? And what's happening to the carbon dioxide? Are we clear? Nice. Now we focus on this intermediate thing, the 6 carbon containing intermediate. Now what happens? This intermediate gets broken down. This intermediate is split from the middle. Now if we split it from the middle, okay, let's remove out certain things over here. Let's erase certain things from here. Yeah, right. Now the intermediate gets broken down from this part. Now from here, if we break down this intermediate, we get two equal halves. One half is this, one half is this, right? Yes or no? The next step, we break down this into two 3 carbon containing halves. Understood? Now, hence, due to the breakdown of an intermediate of 6 carbon, 2 3 carbon containing compounds are produced. 2 3 carbon containing compounds are produced, as you can see over here. 1 and 2 3 carbon containing intermediates are produced. Who's getting removed over here? I'm not understanding. Nobody got removed yet. Now, which one you're talking about getting removed? Nobody is getting removed as of yet. Can you clarify your question? Now, these 2 3 carbon containing molecules, each will contain one phosphate, right? As you can see. Oh, how they are getting, why they are getting divided? Because the enzyme present over there is supposed to do so. And again, another thing that you have to understand, that whenever any chemical reaction occurs in human body or outside human body, what happens? The chemical reactions always chase stability. Now this 6 carbon compound is not stable, so it will try to get stabilized, and hence it gets broken down in such a way so that it can attain maximum stability. Now if you know the idea of potential energy, then you will know the maximum stable or most stable state is known as the state where the potential energy is lowest. That means whenever you have your energy lowest, that stage is your most stable. Yes or no? Right. So that stage is most stable. Now over here also, if the molecule gets divided in such a way, it is observed that the energy of the molecules become lowest, and hence this kind of a reaction takes place. Ultimately in the life, it's all about trying to lower your potential energy or the energy so as to achieve stability, right? Now this molecule is known as 3-phosphoglyceric acid. Right? Now why 3? 3 because over here the carbon 1, carbon 2, carbon 3. To the third carbon the phosphate is attached, and that is why the phospho group is at 3, and the name of this 3 carbon compound is glyceric acid, to which a phosphate group is attached at carbon number 3, and hence the compound name is 3-phosphoglyceric acid. In short, it is known as 3-PGA. Right? Okay, right? Now, this 3-PGA is very important to keep in mind. This is a 3 carbon containing compound, so basically it's a 3C, and this is the first stable product of C3 cycle, or the of this cycle. First stable product of the cycle, first stable compound of this cycle, right? The first stable compound of this cycle. Right? Now, what I told you previously, the naming of these cycles are based on the first stable product of this cycle. And since the compound, the first stable compound of this cycle is a 3 carbon containing compound, the name of this cycle has been given as C3 cycle. Now, can you understand the significance of this 3 in C3 cycle? Exactly, that's why it is C3 cycle. Now, till now, what you are supposed to remember? You are supposed to remember the full name of RUBP, that is ribulose bisphosphate. RUBP is also known as RUDP, that is ribulose diphosphate. That was an older name, right? You are supposed to know RUBP, that is ribulose bisphosphate. You are supposed to know the full name of Rubisco. Okay. Okay. And what you are supposed to know? You are supposed to know the full name of 3-PGA, that is 3-phosphoglyceric acid, and you are supposed to know the carbon containing of these compounds, that is RUBP is a 5 carbon containing compound, 5 carbon. Now 3-PGA is a 3 carbon containing compound. These are important. Now, after we finish off this cycle, I will summarize what are the things that you are supposed to remember. Now, we are coming to that idea of biosynthetic phase. It's not, Calvin cycle can be called, but that is not the entire idea of it. I am coming to that. We will come to that in a while. Within 5-6 minutes, we will come to that, right? And what else you are supposed to know? You are supposed to know that 3-PGA is the first stable compound of this cycle. Okay? Now we move to the next part. Are we clear till this? Can I move to the next part? Okay. So now for our ease of work, let's clear up certain stuff over here. Let's clear up certain mess from here so that it's easier for us to work. Have you noted down whatever has been shown on the board? Okay, I will show you the full cycle at the end of the discussion of the entire cycle. I will show you the cycle. Or should I show you the cycle for a while? You finish it up drawing and then we discuss. Okay, let me show you the entire cycle. You draw this up and then we discuss it. Okay. See, this is the entire cycle, and let me erase out things from here. Okay, now finish off drawing. Just don't put the balancing parts over here. let me omit out the balancings. I haven't discussed those things yet. Draw up the diagram, just omit the balancing parts, right? Don't focus on these balancings. Fast draw the diagram. Don't focus on these balancings. Okay, you're drawing the diagram. Draw the diagram. I'm waiting for you. Once you finish off drawing the diagram, do let me know. I will start. Okay, nice. You have drawn the diagram. It's very nice. Now let's come back to the discussion after PGA, right? Let's start. Now over here after PGA, what happens? We can see, okay, before coming to that, see one thing. This step that is from CO2, to 3-PGA, this step is known as carbon fixation. Carbon fixation. Now this is a very important question that this can come in your examination. What is carbon fixation? Why carbon fixation is necessary? These things. Now what is carbon fixation? I think you can write. You can write. Isn't it? You can write what is carbon fixation, right? Okay, now why carbon fixation is necessary? Why carbon fixation is necessary? Just briefly tell me why carbon fixation is necessary. Briefly. Exactly, that is the step of adding up the CO2 taken in from the atmosphere. Without this step, we cannot add the CO2, the main ingredient of the photosynthetic reaction, the main substrate of the photosynthetic process cannot be taken in to the action, right? This is the process in which the carbon of the carbon dioxide is taken in or taken into the process, right? If carbon fixation is not present, then CO2 will not be utilized in formation of sugar, right? So should we proceed further? Okay, now what happens? We have finished off the carbon fixation stage. Now we will move on to the next stage, that is reduction. Now we had, see, now once carbon is fixed, now this carbon dioxide has to be reduced to form glucose, right? So far we have been able to fix this carbon of this carbon dioxide. Now to produce glucose we are supposed to reduce this, because reduction is the main process, right? Now for that we have this stage known as reduction. Now in reduction what happens? In reduction we utilize one ATP molecule for each 3 PGA. One ATP for one 3 PGA, right? And one NADPH for one 3 PGA, right? These two are utilized, and finally what they produce over here, they produce one compound known as 3-PGALD, whose full form is 3-phosphoglyceric or glyceraldehyde. Do you know what an aldehyde is? Have you started? Okay, have you started learning organic chemistry? Do you know what aldehyde is? Do you know what alcohol is? Okay. Now let me give you a very brief idea about certain things. Okay. Let me give you certain brief idea about organic chemistry. Should I? So this will be helpful when you study organic chemistry in your chemistry classes as well. This is a generalized discussion. Should I continue with that? Okay. So let's go to that discussion first. Now in organic chemistry there are different kinds of molecules. We have certain molecules known as alkanes. We have certain molecules known as alcohols. We have certain molecules known as aldehydes and ketones. And finally we have certain compounds known as carboxylic acids. Right. Now what are alkanes? Alkanes are certain molecules in which carbon-carbon single bond is present, and along with that their hydrogens are bonded. This kind of a structure is known as alkane. Right. These are the very basic kind of organic molecules. We begin the organic study of organic with these molecules. Right. Understood till this? Okay. Now if I remove one hydrogen, let me remove one hydrogen, and in that place if I add one OH group, then that becomes an alcohol. It becomes an alcohol. Right. So can you see over here we have an introduction of an oxygen molecule, introduction of an organic oxygen molecule. Right. So this is a, so we can say that alkanes to alcohol is oxidation. Can we say? Okay, now over here I have drawn another structure. Okay, now over here what you should focus, that over here one oxygen is bonded to a carbon molecule by a double bond. Okay, and with that we have another thing, that is another H group attached. Okay, O2 is getting added. Nice. Now over here O2 had, in alcohol O2 had one bond with carbon, one bond with hydrogen. Now in the new molecule that I have drawn, O2 has both the bonds with carbon. So that also signifies oxidation. This kind of a molecule is known as, if you have this kind of a configuration over here, as you can see, this kind of a configuration in a molecule, this kind of a configuration is known as aldehyde. And from alcohol to aldehyde or ketone is also oxidation. What are ketones basically? Ketones are nothing but, over here if you remove, in case of hydrogen, if you again put any carbon molecule, then that becomes your ketone. This becomes your ketone. This is a ketone group. Got it? Now since in both aldehyde and ketone, oxygen is bonded by double bond to one carbon, they fall in the same category of oxidation and reduction, right? And hence I have written aldehydes slash ketones, right? Now what happens over here, let's take the example of aldehyde. Over here now in this aldehyde, if this H is replaced by OH, if this H is replaced by OH, then that becomes one COOH acid. COOH acid. See over here we have COOH acid, right? Now over here we have addition of one oxygen molecule further, right? Okay, that means this is also oxidation. Isn't it? Now if these steps are oxidation, then the reverse will be what? The reverse in this direction, whatever it will be, this is reduction. Yes or no? Yes or no? Right? Now another thing I should tell you in this connection, that glucose molecule is an aldehyde. It's not exactly an aldehyde, but for your level, from an oversimplified point of view, you can consider glucose to be an aldehyde. Okay. Now the phosphoglyceric acid, so PGA is phosphoglyceric acid, so you have to reduce this acid to get aldehyde. isn't it? You have to reduce the acid to get an aldehyde. Right. So that's why the reduction is required, and once you can produce one aldehyde, you can form glucose from that. Okay. So right now, what is the most important part that you should be remembering for organic chemistry from this class, that your take home message for organic chemistry is this. It's clear, right? So should I go back to photosynthesis? Should I go back to photosynthesis? Okay, nice. So let's go back to photosynthesis and over here. Right. Now over here, you can see the reduction is taking place. Now from here to here, if reduction takes place, then there will be formation of aldehyde. From acid to aldehyde it will be formation. So this is known as 3-PGALD. Okay. Okay. Okay, now since we have produced one aldehyde, our job is almost done. Now what happens out of this? We had two molecules of 3-PGA. Now out of those two molecules of 3-PGA, one molecule will enter in glucose formation. One molecule enters in glucose formation, other molecule will go for one process known as regeneration. Okay. Now the molecule which enters for glucose formation, each 3-PGALD molecule contributes one carbon to the formation of, okay, each 3-PGALD gives one carbon to glucose formation. And the other PGALD goes in regeneration process. In that regeneration process, what happens? So over here, if another PGALD goes, and along with that the rest two carbons over here also go there. Have you understood this or not? Should I repeat this? Should I repeat this? Okay. Okay. Come here. So we had one 6-carbon intermediate, you can write this down, this will be helpful. Now from this what we got? We got two 3-PGA, which are 3-carbon compounds, right? Isn't it? Isn't it? Yes. And now what happened? There was something known as reduction took place. Reduction, and that gave us what? 2,3-PGALD. That is also 3-carbon compound, right? Now what happens? Now out of these two, over here 1 3-PgALD and this another 1 3-PgALD. Okay, it goes into regeneration. Okay, and over here what happens? Over here two things happen. In this 3 carbons, one carbon goes into glucose formation, and the rest 2 carbons go into regeneration. Understood this till this? Okay, now over here we are getting 3 carbon and 2 carbon, so finally forming one 5 carbon compound. Right, 3 carbons from one 3-PgALD and 2 carbons from another 3-PgALD together form the 5 carbon compound. Right, now what is the 5 carbon compound that we saw in the cycle, that we witnessed in the cycle? What is the 5 carbon compound? What is the 5 carbon compound that we saw in the cycle? RUBP, very good. So basically they regenerated the RUBP so that one molecule of RUBP is again ready to take in CO2. So if that is the idea, then we can simplify the cycle as this. We can simplify the cycle as RUBP is here. Now RUBP is again getting regenerated into RUBP here, and what's happening? Somewhere down the line over here CO2 is entering this flow, CO2 is continuing down there, and somewhere else the CO2, one carbon is coming out, one carbon, and RUBP is regenerated. So basically it's a cycle where RUBP is getting regenerated repeatedly, and in this process carbons are taken in, they are getting fixed, and then they are getting channelized into glucose production. Isn't it? Isn't it? Right. Now another thing that we saw, that over here in this place, understand this, in this reduction what happened? In this reduction we saw utilization of 2 ATP, one for each 3-PGA molecule, and 2 NADPH, right? So where we have these utilizations occurring? one over here and one over here. So now if we sum, this is the first place and this is the second place. Now if we sum this up, for one cycle we require 3 ATP molecules and 2 NADPH molecules. Right? Isn't it? Okay, nice. So that is why in every step what's happening? Non-cyclic photophosphorylation. Non-cyclic photophosphorylation will give you ATP and NADPH, but cyclic photophosphorylation will give you only ATP. And that this is the requirement of cyclic photophosphorylation, because ATPs are required in excess number, in more number than the NADPH are required, and hence there are provisions or there are pathways which are there to produce only ATP and not NADPH. That is why we have two alternate pathways for light reaction, that is non-cyclic photophosphorylation and cyclic photophosphorylation. The cyclic photophosphorylation is there to produce the amount of ATP which is required more than the number of NADPH. Basically the cyclic photophosphorylation supplies the surplus number of ATP required. Understood? Okay, nice. So now we come back to the cycle and see what we saw. See, over here we have the regeneration. Over here we have the regeneration process. In regeneration they are utilizing 1 ATP and finally RUBP is getting formed back. So this is the idea of C3 cycle, and similarly we discussed there is something known as C4 cycle. C4 cycle also end up in C3 cycle. Okay. The final common pathway is C3 cycle. In C4 cycle, the first stable product is a 4 carbon compound, that is oxaloacetic acid, that we discussed in the previous class. So now from this C3 cycle is also known as Calvin cycle. Right? Now over here, what are the things that you are supposed to know? You are supposed to know the full form of RUBP. You are supposed to know about, okay, so let me clear up everything over here on the screen. Okay, so I cleared up everything from the screen. Now what you are supposed to know? You are supposed to know about RUBP. You are supposed to know about Rubisco. You are supposed to know about carbon fixation. You are supposed to know about 3-PGA, which is a 3 carbon compound. RUBP is a 5 carbon compound, and this is the first stable compound or intermediate C3 cycle. Right? I'm just summarizing the entire steps. Now in one cycle gives 1 carbon of glucose, right? Now 6 cycles give, sorry, give 6 carbons of glucose. That means for one molecule of glucose production, you require 6 cycles of C3 cycle. This is very important. Right? You're with me? Now what? For one cycle, you require 3 ATP, 2 NADPH. So 6 cycle, you require 18 ATP, 12 NADPH. Okay, I'm waiting. Should I continue? Should I continue? Okay, nice. So now, so 6 cycles, so one molecule of, for formation of one molecule of glucose, you require 6 cycles. So this can be a question for one molecule of glucose, how many ATP molecules and how many NADPH molecules you require. So that you can answer from now, this idea you can answer. So whatever I have written right now on the board is important, just please note it down on your copy. Whatever I have written over here on the board. Right? Should I proceed further? Should I proceed further? Now, few minutes back you asked a question. So is Calvin cycle the biosynthetic phase? Now let me answer that question for you. Now, the anabolic reactions are known as synthetic reactions. Right. Now, as in the dark reaction or light independent reaction, the proper anabolism takes place, and anabolism also means most of the times reduction. Since the proper reduction of carbon occurs in the dark phase, a dark reaction or dark phase, it is said that the proper anabolic reaction also, the main anabolic reaction is in the dark reaction. Light reaction is just a preparatory phase for this anabolism. And if anabolism is occurring in the dark reaction, synthesis or synthetic reactions are occurring in the dark reaction, we can say, since anabolic reactions are essentially synthetic reactions. And hence that is why the dark reaction is known as dark phase is known as biosynthetic phase, not the Calvin cycle. Now there are certain plants for whom C3 cycle or Calvin cycle represent dark reaction. For them we can say Calvin cycle is a biosynthetic phase. So can you answer this question? There are certain plants for whom C3 phase or C3 cycle or Calvin cycle represent the dark reaction or dark phase. For them, C3 cycle is the biosynthetic phase. So basically dark phase over here, not reaction, phase. And with this we wrap up the chapter of photosynthesis. Any questions, or should I terminate the stream? In the next class we will do what? In the next class we will do plant hormones. Okay, so let's end the stream over here. With this was a very big chapter, very conceptual chapter. It took very long, but finally we are done with this chapter. In the next class we will start with plant hormones. Right, so let me end the stream over here. C4 cycle you don't have in your syllabus. Just remember that the first stable product of C4 cycle is oxaloacetic acid. That's it, nothing more than that. There are certain plants who have C4 cycle, and for the C4 cycle the first stable product is oxaloacetic acid. I discussed that in the previous class in the introduction of the dark reaction, right? All right. Actually we don't have the entire details of the C3 cycle as well, but still I covered the C3 cycle so that you can answer these questions. And C4 cycle is not at all in your syllabus, that's present in your class 11 syllabus, right? So now let's terminate the stream. Bye, see you in the next one.